Hi, I'm Matt Peterson from the MITRE Corporation, and I'm going to talk about our application of natural language processing for extracting COVID-19 drug and vaccine information from scientific literature. With emerging diseases like COVID-19, there's an urgent need to stay up to date on treatments that have been tried and their associated results. During the outbreak, more than 1,000 journal articles, preprints, and clinical trial records are being published per week about COVID-19. We set out to help the biomedical community more easily find scientific information about drugs and vaccines for COVID-19, as well as related diseases like SARS and MERS. To do this, we developed a natural language processing pipeline to extract drug and vaccine information for SARS-CoV-2 at scale from the rapidly evolving literature and make it publicly available via a web-based dashboard. There were several challenges we faced in developing this pipeline. We needed to rapidly develop and deploy it due to the nature of the task. There was also little to no available curated data for training a system at the start of the outbreak. Finally, we needed to build a system that was adaptable to the rapidly changing landscape of available literature. Here, we show the overall approach we took to solve this problem. First, we retrieve relevant articles using the NCBI APIs, preprints from the COVID-19 corpus, and clinical trial narratives from clinicaltrials.gov. We used a rule-based NLP system, which I'll discuss in a bit more detail next, to extract information from sentences at scale. We then leveraged redundancy at the abstract level along with contextual information to prioritize these results. The results are then loaded into a web-based dashboard as well as a curation interface where human-machine teaming is used to further improve the results. This pipeline is run every few days, capturing new papers, and new results are integrated with the previously extracted information. Next, I'll go into a bit more detail about the extraction steps in the pipeline. To extract information from papers, we utilize a two-stage extraction process. In the first stage, we use the REACH system developed in the Computational Language Understanding Lab at the University of Arizona to extract entities, relations, and events at the sentence level. In the second stage, we assemble this information extracted across sentences at the abstract level. Entity extraction is largely dictionary-based, using both standard biomedical resources such as DrugBank, UniProt, and the NCBI taxonomy, as well as custom dictionaries. Relations and properties use a combination of surface and syntax rules to identify relationships between these entities. These relationships include proteins that are identified as text as entry receptors for a virus, vaccines tested or used to protect against a virus, drugs used to treat a virus, and the specific vaccine type, for example, mRNA or protein subunits of a mentioned vaccine. In addition to these entities, we also identify key indicator phrases associated with the current stage of research, whether the work is being done in silico, cell lines, a model organism, or clinical trials. Here we see an example phrase, a purified and activated SARS-CoV-2 virus vaccine. We extract three entities, the vaccine type, vaccine, and virus. We also identify two relationships, the relationship between the vaccine and type, and the relationship between the vaccine and virus. Next, we utilize redundancy and context to assemble the results at the abstract level. For each abstract, we calculate the incidence and co-occurrence of events, entities, and indicators in the title and abstract. We calculate evidence scores, which are weighted sums of these factors. Abstracts are assessed as being about a virus and one or more of a drug, vaccine type, or associated entry receptor if they meet a minimum evidence score threshold. Here we have an abstract that our system identifies as being about an inactivated vaccine for SARS-CoV-2 that has been tested in non-human primates. Once the results are assembled, they are displayed in our web-based dashboard we call the COVID-19 Therapeutic Information Browser. The browser will be demonstrated in more detail during the text mining COSI demo session by my colleague, Tanya Corvus. The browser summarizes information about virus drug, virus receptor, and virus vaccine type pairs. Each cell contains the number of abstracts where this information is found. Here, the circled cell indicates that there are 139 papers discussing the use of azithromycin in the treatment of SARS-CoV-2. Users can sort this table, search for a specific therapeutic, and filter the results by date. When a user clicks on the cell, they see a detailed list of the papers summarized in the summary table. This includes the date of publication, the source, and the article title with a link to the abstract, preprint, or clinical trial record. If the paper was curated, the research stage and efficacy assessed by the curator is shown. For curated papers, sentences supporting the relationship between the virus and the therapeutic or entry receptor are also shown. In this example, the curators highlighted the paper's assertion that the compound tested was shown as effective in the sentences they chose as evidence. To date, we have processed over 86,000 abstracts and clinical trial narratives and extracted virus therapeutic-related information from over 8,000 of those. We've identified nearly 1,500 candidate drugs across the 20 viruses we have read for, 188 of these for COVID-19. This browser has been accessed by users all over the globe. We continue to update our pipeline and dashboard. In the near term, we plan on linking out to additional information such as adverse effects and proposed mechanism of action. We also plan on capturing new drug vaccine candidates as the literature grows. Finally, we would like to develop validation data sets to assess the performance of our system and document our lessons learned for future scenarios where such a system may prove useful. We're also interested in identifying new use cases and applications which our browser could support. So if you have any ideas, please contact us at the email address listed on the browser's homepage or the poster and get in touch during the poster session. Thank you for taking time to watch this presentation.